everyone, welcome back to my YouTube channel, Lisa Does Science. Today I'll be talking to you about vaccines, specifically against the virus that causes COVID-19 disease, and this virus is known as SARS-CoV-2 virus. So first of all, before we start talking about the vaccines and how they work, let's revise the structure of this virus because we need to understand what the virus is composed of in order to understand how the vaccines work. So first of all, you can see here that the virus and most viral structures are quite simple. Most viruses have an outer envelope and a lipid membrane that separates the virus from the rest of the environment. So this particular virus, SARS-CoV-2, has a genetic code inside that is not in the form of DNA. It's actually in the form of RNA. So what is RNA? RNA has a very similar structure to DNA, the only difference being that it's normally single-stranded. So unlike the double-stranded DNA helix that wraps around itself, RNAs only have a single strand. So why is it important that this virus has RNA? It's because according to the central dogma of molecular biology, in order to create a protein from the genetic code of the cell, which is normally DNA, like in our cells, it has to go through different stages. So there are two different steps involved in this process, and originally DNA gets transcribed to RNA inside the nucleus of the cell. Okay, and then the RNA can leave the nucleus and travel into the cytoplasm of the cell, where it then gets translated into the protein. So this is very key for us to understand in order to understand how many of those vaccines work, because this is the first time in history where an RNA vaccine has been put into market and in the population. So this virus has a single-stranded RNA as its genome, which means that it only requires one step between RNA and protein to produce the protein that are required to assemble the virus back. So what are the proteins that this genome actually encodes for? Okay, there are only 4 proteins that are required for this virus to exist and survive. So we have a nucleocapsid protein that actually packages this RNA inside the virus. We also have a membrane protein, an envelope protein that I've talked about before, as well as the spike protein. So the spike protein, you would have probably heard on the news before. This protein is extremely vital for this virus because this is exactly how the virus gets inside our cells. So how it works is that the spike protein has a receptor binding domain. Specifically, it can bind to this receptor called ACE2 receptor. ACE2 standing for angiotensin converting enzyme 2. So our cells normally express the receptor. I won't go into details of what this does, but normally this receptor is expressed on the surface of the cells. So this virus's spike protein can actually recognize and bind to this receptor, and this is how it gains entry inside the cells. Remember from your biology lessons that viruses are sort of this very close to the life form, but some scientists still argue whether it's a life form or not, because actually viruses can't survive without other living cells, so they can't replicate themselves outside of another living cell. So therefore, entering the cell for this virus is the step that is absolutely required for its transmission and for its prolongation of its lifespan by replicating itself and creating copies and copies and copies of this virus inside another living cell. So I hope this is clear. And now that we have visited the structure of the virus, we can now move on and talk about the vaccine specifically. So the way I'm going to tell you about the vaccines is I'm going to split them into 3 groups. The first group is going to be the RNA vaccines, so the ones that I think people have heard about the most on the news. The second is going to be a more conventional weakened virus sort of vaccines, such as AstraZeneca and Oxford one. So this is going to be the second category of vaccines that I'll tell you about today. And the third is going to be a completely different vaccine that you might have heard of recently, which is a Novavax vaccine, and I'll tell you about that specifically in more detail. And the reason I'm putting it into its own category is because it doesn't fall into the first 2, and we'll talk about why later. So the first category are the RNA vaccines. So we are living in a very exciting moment in time where, for the first time ever, RNA vaccines have been licensed for emergency use. So this is really, really exciting, and the reason for that is that those vaccines can be created to treat other diseases as well, potentially, such as cancer. So why do they hold advantage over the other more conventional vaccines that usually use a viral particle or a weakened form of the virus? Is because they only require RNA. And as I already told you, RNA is just another form of genetic code, similar to DNA. So the reason for its advantage is that it's very easy to modify this vaccine, so change the letters of the code that are required to produce the protein. And in the lab, you can get an experimental batch of vaccines in only 3 weeks' time by changing the RNA code. And of course, this is really important, especially now that we are seeing so many new variants coming out, such as the UK variant, such as the Brazil, a Portuguese variant, and others, and South African. We really need to have a technology in place to potentially have to change the vaccine according to the new variant, such as what the flu vaccines do. Normally, you would get a flu vaccine every year, and sometimes they get modified depending on the prevalent strain. So I hope this is clear. And of course, the RNA vaccines against the SARS-CoV-2 virus have been produced by 2 companies in collaboration with Pfizer and BioNTech company in Germany, and also Moderna. And both of them have a very superior efficacy as judged in the clinical trials phase 3 over all the other vaccines that I'll tell you about. And the vaccines actually come in 2 doses. So the clinical trials have only trialed the interdose interval of just 3 weeks. So you're getting your first dose, and then 3 weeks later, you opt to receive your second dose. However, in the UK, the government has decided to prolong the period between the doses for up to 12 weeks. And the reason behind this was the fact that you're trying to get as many people as possible to get vaccinated with just the first dose that actually provides the highest efficacy anyway. So the government decided that by increasing the total number of people vaccinated would be more beneficial for the population overall than getting the 2 doses in a very short period of time, because it is very likely that the first dose would actually confer the most protection. Right, so how do the mRNA vaccines work? And what is mRNA? So mRNA actually stands for messenger RNA. And this is the messenger that tells our ribosomes inside the cells to make protein. So exactly by intramuscular injection in the arm, the vaccine is then enters the muscle cells within your muscles and then it is able to make the spike protein. And once the spike protein is made, it gets displayed by your own cells for the immune cells to recognize the foreign antigen and then start producing T cells and B cells in response. So T cells and B cells are just referring to two different types of immune cells, and B cells are actually the ones that will go on to make antibodies that match this antigen, and they're able to come and neutralize it. So this is how it works on the surface. And the reason why a lot of people are getting worried right now because of the new variants coming out is because those variants are potentially changing the structure and the shape of the spike protein. So a lot of people are getting worried whether the antibodies are still going to do their job. But Pfizer has already come out and said that their preliminary data indicates that actually their vaccine is still effective against the new British variant, for example. So the reason for this is because when your B cells produce the antibodies against the antigen, they're not just producing one type of antibody, they're actually producing loads of different antibodies that are able to recognize different places of the antigen. And this is called the epitome. So the epitome is the specific stretch of your protein that your antibody binds to. And the immune system has developed the tools that it needs to create many different antibodies, again, to protect your body from this virus mutating and potentially having slightly different structure of its protein to what it was when you got vaccinated originally. So I hope this explains why the vaccines might still be active against new variants. However, if the virus accumulates a lot of mutations consistently, that's when you might encounter a problem. It is heavily monitored to ensure the safety and the efficacy of the vaccines at the moment against new variants as well. And I'm aware that there are some trials ongoing in South Africa as well. So now, the main disadvantage of those mRNA vaccines is, of course, the fact that because RNA is such an unstable molecule, you need to really store it at a very low temperatures. So unlike DNA, we always hear in the news, oh, the DNA of a really old, for example, old organism has been found and has been sequenced. And the reason for that is because DNA is really, really stable over time. Again, because it has this double helix, it's protected from degradation in a way, but mRNAs are single-stranded and they're usually shorter sequences, so it's actually a lot easy to degrade RNA. And therefore, those vaccines are normally stored at -70 degrees, which really makes it difficult to transport the vaccines and store the vaccines because you need to have a lot of freezer facility and storage for -70. All right, let's move on now into the second category of vaccines. So the Oxford and AstraZeneca vaccines are using weakened common cold virus called Adenovirus that only causes cold in chimpanzees. So this is why the vaccine is called ChAd-Ox, because it's chimpanzees Adenovirus Oxford, and they've used the adenovirus that's been modified to only cause common cold symptoms in chimpanzee monkeys, and this is only acts as a vessel, sort of as a vehicle to transport the genetic material inside this virus into the muscle cells. It can be stored at higher temperatures than that of mRNA vaccines. So it can actually be stored in the fridge. So it gives a massive advantage for logistics of transporting and storing the vaccines, especially in the amounts that they're required to protect populations that have millions of people, such as UK. So the more conventional vaccines, such as AstraZeneca and Oxford, contain a weakened adenovirus that has cDNA inside of it of a spike protein. What is cDNA? cDNA just stands for complementary DNA, and it means that you can synthesize your complementary DNA from your single-stranded RNA sequence of the virus. You then sort of go backwards to generate DNA, and then you insert the sequence into your vehicle, which is adenovirus virus. There are two other vaccines that fall in the same category. The first one is Sputnik V, which is a Russian vaccine. Again, it uses adenovirus. Actually, they're using two different adenoviruses inside of it to act as a vehicle, and they use cDNA of a spike protein inside of it. And this has actually been the first vaccine in the world to be used, but it has been met with a lot of controversy because the Russian government decided to use this vaccine only after phase 2 data, so phase 2 clinical trials, and as we know, other vaccines had to get approved only after completion of phase 3 clinical trials that require dozens of thousands of participants to undergo this. So this is a very rigid and very highly regulated process of approval of any drug or any medicine used. And then there is another vaccine that only recently has been bought, and it's a Johnson&Johnson vaccine. Again, it uses an adenovirus, and it has a slightly lower efficacy than the AstraZeneca, Oxford, and Sputnik V vaccines. It only has 66% efficacy in preventing symptomatic disease. However, it's 85% efficacious in protecting against severe COVID-19 disease. And I believe that Sputnik V has an efficacy of 91% reported from interim data of phase 3 trials, putting it in the third place after Pfizer and Moderna vaccines. And AstraZeneca and Oxford had slightly lower efficacy. However, on the 3rd of February 2021, AstraZeneca's website said that they had 100% protection against severe disease and hospitalizations and death in primary analysis of phase 3. And they also very importantly showed that after taking this vaccine, the rate of PCR positive tests for the virus has decreased by 67%. Therefore, it's evidence to suggest that not only it's efficacious against symptomatic and severe disease, it can actually decrease the rate of transmission, which is so essential when we're trying to stop the spread of the virus. And by stopping the spread of the virus, you're also reducing the rate of novel variants emerging. And it's extremely important to consider because only yesterday, the Queen of England has spoken about the vaccines, and she said that people who are reluctant to take the vaccines or who are hesitant to take the vaccines should really think about others and not themselves when it comes to it. Because if you think that, you know, you're unlikely to suffer from severe disease due to your age or due to your medical history, still, please consider taking a vaccine in a safe environment, because that would mean that you would actually be contributing to a reduction in rate of transmission of the virus. Not necessarily in 100% of cases, but AstraZeneca has shown that by 67%, the transmission has been reduced. Vaccines have actually been around since 18th century, and England was the first country in the world to make the first vaccine. Edward Jenner made vaccine using virus that causes cowpox in cows, and then administered it and showed that administering cowpox virus was effective against smallpox. And in just 2 years' time, they were ready to produce these vaccines after he showed that. So after just 2 years, the vaccine against smallpox was available in England in 18th century. So people who try and say that, in 2021, it's impossible to have a vaccine just after 1 year, you must realize the effect of this global pandemic that it's had on economies, trillions of dollars lost, and by investing billions of dollars into development and testing of these vaccines is really nothing in comparison to what we lose as global economy. So you have to understand that the vaccines were first created more than 220 years ago. And now let's move on to the third category of vaccines, which is completely novel and quite exciting. And this vaccine is called Novavax, and it actually uses a completely new technology, which the producers of these vaccines have branded as a recombinant nanoparticle vaccine. And the reason they call it that is because they're able to make the spike proteins in the lab. So those are just the spike proteins without the virus on their own. And what they can do is make a synthetic lipid sphere that has the spike protein inserted into the lipid membrane. And this lipid nanoparticle is only about 50 nanometers in diameter, and this is why they've called this a nanoparticle vaccine, because it uses a lipid sphere that has a nanometer sort of range. So each lipid sphere can display up to 14 spike proteins. So when the Novavax vaccine is taken, your body recognizes the antigen, the foreign antigen straight away. So it doesn't actually have or carry any genetic code in it whatsoever, either DNA or RNA. Preliminary efficacy data has shown on the 28th of January that this vaccine has 89% efficacy. So this is really exciting news. And again, another technology being used, although previous technologies similar to that have been used in the past, and normally those are just a subunit protein vaccines. So they carry a protein or a part of the protein that causes that is present in the virus, for example. So again, this is based on old technology, but this is slightly different. So again, we are having this massive technological progress, and it's very exciting to see, and I'm really hopeful that the vaccines would help change the course of this pandemic. As we already know from the data from the UK, from middle of January till middle of February, the rate of deaths in over 80s, which were the first group to be vaccinated by age, over 80 years old, has been declining, and has declined significantly over this time. And I'm very hopeful that the vaccines would change the course of the pandemic. This is everything from me. I hope this video has been really useful for you. Please leave a comment down below if you want me to make a similar videos like that, or if you want me to make any biology related videos that you might want to learn about. And don't forget to like and subscribe, and I'll see you in the next one.